Thank you very much for the good introduction. So you all know where I come from and what the type of work that I do. And I'm actually very happy to share with you the research that we do and trying to have smart data in order to unravel pharmacokinetic and pharmacodynamic relationships with the focus on the central nervous system. So the question that we ask ourselves is actually how to predict human CNS drug effects in health and disease. And this is actually the scheme that I use all over, always, because we have drug dosing, then we have plasma pharmacokinetics with the focus on the unbound pharmacokinetics, and then we have the blood-brain barrier. I will tell a little bit more about that. And then in the brain, we have the unbound brain pharmacokinetics, especially then at the target site. Then there is target interaction, binding kinetics. Then there will be cellular transduction mechanisms, ultimately leading to an overall effect. And this overall effect will also have kind of homeostatic feedback because the body tries to get back to its original state. So there are all kinds of processes ultimately between drug dosing and effect and how to deal with that. But first, for those who are not so much into pharmacokinetics and pharmacodynamics, just in short, we can have a concentration as a function of time. We can have different profiles. And actually, the best profile is, of course, the ones that is mostly in the area of concentrations where we do have the favorable effect. If it's too low, okay, we do not have any effect. If it's too high, we have more side effects and more adverse effects. So we need pharmacokinetics in order to get to the best pharmacodynamics. I always like allegories, and this is an allegory of that you might think of. So in the body we have interconnections and relationships, and I try to have the allegory of a driver and a car. So if we would have Max Verstappen, I'm from the Netherlands, right? If we had Max Verstappen and he drives in his race car, he has excellent car performance. If I would be in this race car, it would be completely different, I think. And also, if he would be in my car, then it would also be different. So we have actually a combination of a driver and a car that determines what comes out. But also, we can have this allegory as we have a drug and we have a certain system. And the combination of the drug and the system determines the pharmacokinetics and the pharmacodynamics. So pharmacokinetics are not drug properties. Pharmacokinetics is the result of drug properties and system properties. Let's put it like this. So we have drug properties, we have system properties, so rat or mouse or human beings or patients or whatsoever. And they combine in a result in pharmacokinetics in plasma, but also of course at different places in the body, and also the pharmacodynamics. Well, if we have a drug, the drug remains to have its properties wherever it's been given to. So if I receive the drug has its properties, and if a patient receives the drug still has its properties. If we give it to an animal, the drug has still its properties. But what is actually variable is where we combine it with the system. Either it is the rat, the mouse, the patient, the human being, etc. So we have all kinds of physiological parameters like put here, so blood flow, barrier permeabilities, transporter, enzyme functions, volumes in the intracellular space, extracellular space, pH in different compartments, surface area of capillaries or membranes, receptor expression, etc., etc. So actually what we need to do as the basis for translation is to explicitly distinguish between drug properties and system properties. So here you see it depicted, and again always all the mechanisms between drug dosing and the effect. So this is for an animal, and you have a certain rate and extent of all these mechanisms in between. And if we have differences in system properties, like I explained before, coming from the animal, going to the human, for instance, we know that that might result in differences in pharmacokinetics, blood-brain barrier transport, binding kinetics, and pharmacodynamic processes. And I will get to that later as well. But translation needs to be done on the basis of information of all these steps in between. That is the key message. So smart data. So this is a full slide with a lot of text, so I will go through. Well, if we talk about the central nervous system, we know that effects are not easy to be interpreted. It is quite complex and it's controlled by many mechanisms, and we have variabilities between animals, between humans, patients, etc. So CNS research is complex. And we all know, at least I hope, that we cannot sample the human brain. You can have non-invasive techniques, of course, but definitely not invasive ones because of the ethical rules, of course. It means that we need to find a way to get this information indirectly. And we have learned that the rate and the extent of all the mechanisms between dose and ultimate effect are context dependent, systems dependent. So we need to have animal experiments actually to be able to see what the contributions are of each of these steps. And that is then the basis for translation to human. One important information is time course information, because at some point in time, it's only limited information because things may be quite different at another point in time. So we need time course information from subjects at multiple levels because everything is connected. And now I come to the mastermind research approach. In the next slide, I will discuss this little game. And actually, before doing so, I like to emphasize that connected data, if it comes from one subject, can be really informative. And with the combination of mathematical modeling, you can really get the rate and the extents of the processes in between. And mathematical models are actually very important to organize, condense, integrate, and also store knowledge and understanding. So you really build a house from all building blocks of knowledge. What a slide. Okay, let's go to the game. Whom of you has ever played the game Mastermind? So that is quite a bunch, not everyone. Okay, so I explained it shortly. So you have two players, one on this side and one on that side, and this is the board in between. This player makes a hidden code of pins with different colors and different positions. And the task of player 2 is actually to unravel the code. And it's important to notice that you have a position, so there is an order and a different intensity by the color, so that is the allegory. Okay, so what should you do to unravel the color? You should not investigate only one position of one pin, because then you have not enough data to understand what the code is. So it means that you have to do on multiple levels, so here, and try and then strategically combine and use the feedback of this player, because he says here, for instance, yeah, there is one pin that has the right color in the right place, so that is on this position. And here, changing that one, okay, it's the right color, but not in the right position, etc., etc. So you get feedback, and with that, you kind of learn actually how to tease out ultimately the right code. And it's always possible in this 6-numbered mastermind game, if you do your best. Okay, so it actually indicates that we should not focus on one aspect, but we should investigate things together in order to really understand what is happening. So that is the reason why I use the allegory of the mastermind research approach. And then we come to the blood-brain barrier. So we know the central nervous system, and it has a huge brain capillary network depicted here. This is a cast of a human brain. And if we zoom in, we see individual capillaries. And here you can see a cartoon of a capillary. So here you can see the blood, and this is actually the size of a red blood cell. So to give you an impression of the size. Here we have the brain cerebral endothelial cells encompassing this capillary, and also there are kind of end feed of astrocytes, so typical kind of brain cells that kind of influence the functionality of these brain cerebral endothelial cells. And if we go to transport of drugs out of the bloodstream into the brain, we can consider this next cartoon. So here the brain cerebral endothelial cells are depicted, and actually what we can see is that we have spaces between the cells that are narrow. So these are the tight junctions. So this makes that the traffic here is limited, because not so many drug molecules can fastly go to the other way because it's so small. One by one they need to go. This is called paracellular diffusion, and it's passive, and especially drugs that are hydrophilic need to go through this route because they cannot diffuse across membranes because they are just not lipophilic enough. And if you have drugs that are more lipophilic, they can diffuse over the brain cells. That is both passive transport modes. Then we also have active transport modes. So always from one side to the other side. from plasma into the brain or from the brain into plasma. So influx transport, efflux transport from this way to that side. And there are also vesicle-based transport modes, but these are more important for the larger molecules and the biologics. And I will not focus on that, so I will focus on the small molecules, and the paracellular diffusion, transcellular diffusion, influx transport and efflux transport are important for the small molecules. So that is what we then take into account. So this blood-brain barrier, is it important or isn't it? So here I just have a kind of example of a concentration of a drug in plasma. So this is the unbound concentration as a function of time on the logarithmic scale. So then it looks like this, if you have a simple one. And if we consider only passive transport, then things in the brain may be quite different between the ones that have fast blood-brain barrier transport. Because if it's really fast, then actually the brain concentrations, the unbound brain concentrations, kind of are the same as the unbound plasma concentrations. And that is for some barbiturates. Some drugs indeed can have it like that. But if you have slow transport in and slow transport out, all the processes lag behind. So it takes a long time to reach the same concentration in plasma. And if it's so here it rises, then it decays also very slowly. And then you can see that you have a very distinct profile in the brain extracellular fluid relatively to what is in plasma, indicating that plasma cannot easily be used for interpreting what happens in the brain, because this is not the only thing that happens there. Okay, so what is the conclusion? If we are to understand CNS drug effects, we really need to understand drug distribution into the brain, how these profiles look like. So brain extracellular fluid concentrations or intracellular fluid concentrations even are very important. Also, time course of drug concentrations is important, and especially knowing unbound concentrations, because it's the unbound concentration of a drug molecule that can be transported or can diffuse, and also it's the unbound concentration that can bind to targets, right? So that is kind of the most informative part. So that is actually crucial. So these unbound time course concentrations are crucial to understand the rate and extent of transport processes and also of binding kinetic processes. Well, there is an important technique that is able to monitor drug concentration, so to have this time course. So that is actually the microdialysis technique. So just in short, we have a microdialysis probe, and it's actually so tiny so that you cannot see if I do like this. And it is the tip of the microdialysis probe that has a semi-permeable membrane. And you can flush the microdialysis probe. And if we then zoom in at the tip of this microdialysis probe, so there's fluid going in and fluid going out. And then at this tip, small molecular weight molecules can cross the membrane, because they are small enough for this semi-permeable membrane. And they can exchange, and they can be taken with the fluid flow, and the fluid can be collected in intervals, and we can measure the drug concentration or any other compound in this collected fluid, and then have concentration time courses. And that may be of drugs, but of course also of potential biomarkers of drug effects and or disease. So it means that you can collect quite a bit of information from one animal, and that is important. And it's connected data, because this animal has, for instance, this plasma concentration in red, this brain extracellular fluid concentration in blue, and this EEG profile. So it's connected. You're sure it is all happening in that same animal, and that is important as well. So microdialysis is a key technique in order to link the drug concentrations ultimately to effects in terms of time course. And now I go to a first example. And this is the work that has been done by Jasper Stevens, a PhD student in our lab, longer time ago. And we were actually looking at how can we have information on the dopaminergic system in the brain of human using a biomarker that can be measured in plasma. Well, we know that in the brain we have multiple dopaminergic systems, and one of them is coming from the hypothalamus and feeds into the pituitary, And in the pituitary, we have the so-called lectrotrophs. And in these lectrotrophs, we have little vesicles where there are hormones. And for instance, in the posterior pituitary, we have prolactin in these vesicles. And prolactin release is actually under control of the dopaminergic system. Because if dopamine is high, then prolactin is kept in the lectrotrophs. If dopamine is low, there is a release of prolactin into the bloodstream. So we can measure in the blood what's actually happening here. But also, if we use dopamine D2 antagonist in this case, we also induce a release of prolactin. So this is the paradigm that we used, and we try to work with animals in order to understand all these processes and translate it to human. So what we did is we gave remoxapride in 3 different dosages. So the low, the medium and the high. We measured with microdialysis the extracellular concentrations in the brain of remoxapride, like here. And also in plasma, because it's released in plasma, prolactin can be measured in plasma. So we also had the time course of prolactin there. And actually we did this after intravenous administration as well as after intranasal administration. Also, we can translate between those two. But in real life, if we would have a patient, of course, we will have repeated dosing. And what would happen if you have repeated dosing and first dose releases more or less all prolactin? But how long does it take to restore the original situation again? And then really see how fast this process goes. So that information is also part of this pharmacodynamic modeling. And to that end, we had the double dosing design. So we had the first injection and the second injection shortly close to each other, or more lengthy, or even more longer, or starting at a later part of the day because of circadian variability, and also have a 2-hour, a 4-hour and a 6-hour interval, and just see what would happen with our prolactin concentration in plasma to understand how fast prolactin can be restored in the same situation to have the original one back again. Okay, so having this information, and just to summarize shortly, we have intranasal administration, then we have also plasma concentration, because via the nose it gets also into the bloodstream, but also into the brain. That is what we show here. This is the brain extracellular fluid. Here we have the intravenous administration, plasma concentrations, and the intravenous brain extracellular fluid concentration. And on this basis, connected to our prolactin data in plasma, we could build a pharmacokinetic pharmacodynamic model. And pharmacokinetic, so we have concentration in plasma, it's called the central compartment here. We have it in brain microdialysate, so the brain extracellular fluid, and a lumped peripheral compartment. And we needed 2 absorption pathways, one from the nose directly into the brain and one from the nose into the bloodstream. Both occur, and we needed both because we did a lot of other models and tried to see how we could best fit the data. And here you can see the quality of the data fitting. It's really capturing all the data. And then for the pharmacodynamics, it's important to realize that what we actually measured with brain microdialysis was directly the target site concentration, because there was no delay between the profile of brain extracellular fluid remoxapride and the effect. So it means that you're really at the target site. So that is an important thing you can only measure in animals this way. So remoxapride ultimately has an influence on the release of prolactin from the lectrotroph. So it releases into the bloodstream. So we have a certain concentration of prolactin in the bloodstream. And if this concentration actually has a positive feedback on the synthesis of prolactin, just saying, okay, this lectrotroph has been depleted, please restore, speed up. That is actually the homeostatic feedback. Okay, this is in animals. And then we try to have the same model structure applied to human. And we kind of translated individual things that we could do on the basis of in vitro values for humans compared to animals, and also allometric scaling of plasma pharmacokinetics, etc. Actually, I can show you the results. So this is the pharmacokinetic part of the model. And here you see individual animals. All boxes is an individual animal. And you see the open dots that are the data, and you see the model nicely capturing the plasma pharmacokinetics of rimoxapride in animals. And we had a data set from Uppsala where we collaborated with, and here you see the collection of a double dosing design of rimoxapride in human beings, and also the model was very nicely able to capture that with the same structural model. Then the pharmacodynamic part of the model. So here we have, again, the red, we have prolactin, here displayed in the double dosing design, so you can see that the model nicely captures actually what is happening there. And here you can see how, after translation from rat to human, how we could also capture the double dosing design prolactin effects in human. So here we had a very good translation. In human we needed the link in the brain. That was provided by the animal. By translating from animal to human, we could make this link also in human and understand dopaminergic activity in the human brain by this. So that was actually a very nice exercise, I should say. For this, in total, to make this whole model work, we used the smart data, concentration, time profile, different locations, etc., from only 23 rats to be able to do this, of course, in combination with mathematical modeling. And this is an example for one individual drug. And I indicated that the pharmacokinetics in the brain extracellular fluid is actually very important, because that is kind of facing most of the target sites. So then we asked, okay, in human, could we have a general approach in order to predict these target sites in human brain? Because mostly, if we are interested in target site concentrations, actually CSF concentrations are used. Mostly from the lumbar region, where my wire is now, you know? So it is quite a distance, you can say. So I questioned whether this concentration here would be equal to this concentration or that concentration. So we stepped into that. And then I come with the TomTom. Who knows the TomTom? Many of us use the TomTom. I want to drive to this. Okay, how long does it take? La-di-da-di-da. For being able to have a TomTom, they needed to have location and time dependency, because I show you here the Netherlands, the map of the Netherlands and main routes, and actually the density of the traffic, let's put it like pharmacokinetics, so here it's high concentrations relatively, at this point in time, so this is not in rush hour, but if you see this picture and then later, what do you see? You see changes not only at one place, but at multiple locations. And it's of course different. So you have location and time dependency. And if we look at our brain, it is actually the same, I would say. So location and time dependencies is what we need. And this was actually the way that we started with, okay, let's make the map of the brain. So we started with kind of depicting the physiological CSF compartments. The blood-brain barrier, the blood-CSF barrier. I won't go into detail, but that is this part. Then we have the brain parenchyma with the brain cells and the brain extracellular fluid. And we have multiple compartments of the cerebrospinal fluid, the CSF. We have the lateral ventricles, the third and fourth ventricles, the cisterna magna, and then the spinal cord, and the lowest part is the lumbar region there. Okay, so this is the map. Then what do we have? Okay, we have fluid flows, because fluid also takes molecules. So we should take that into account. The blood-brain barrier produces fluid, and it goes ultimately in the direction of the cerebrospinal fluid. And also the blood-CSF barrier produces CSF, and ultimately it is really put back into the bloodstream again. So this should be added to the map, to the information. Then we have surfaces of membranes. So the brain cells do have a certain surface area, and also the blood-brain barriers. We have membrane properties, so there can be passive transport across the blood-brain barrier, across brain cell membranes. We can have, of course, also active transport, not only at the level of the blood-brain barrier, but also at the level of the brain cells that we need to take into account. So the active transport part. And we also have enzyme functionalities, because some drugs are really converted in the brain, so it can be extracellularly, it can be intracellularly, but sometimes even at the blood-brain barrier. So that is kind of all the aspects that we need in terms of a map. And then last but not least, we also have important subcellular compartments. And that is important because these subcellular compartments especially have a very low pH. But also you need to take into account the pH values of the other fluids. Because if you have drugs with ionization constants, the ratio between the charged and the neutral form depends on the combination of their ionization constant and the actual pH. So that is taken into account. Then, okay, if we have that, we know that if we have a drug and we put it in this system, we will have resulting pharmacokinetics. And I said location and time dependency. So how do we measure at multiple locations? Of course, we cannot do that in human brain. We cannot access that with this technique. And this technique actually gives only the CSF, but we can have the experimental animal that could serve as a bridge between those two. And that is where we started. So what we did, and here again you see the full map with all the processes that occur and the 3 microdialysis probes that actually have been put. So this is a cross-section of the rat brain. So one in the striatum and 3 different locations in combination with also serial blood sampling. So what happened? And I should, by the way, here indicate Joost Westerhout, who did these experiments, a PhD student also. So we had an animal experiment, and now I have the example for acetaminophen or paracetamol if you wish. So 3 locations in the brain and serial blood sampling. And we had concentration time profiles, so you see the data points from these 4 locations. And as I indicated before, based on the physiological map, we made a structural physiology-based model. And it looks like this. And I depicted some typical values of a flow and things like that. Or brain extracellular fluid is, for instance, 290 microliter on average in animals. And CSF flow is about 2.2 microliter per minute. Well, actually, if we use this model and kind of see if we could model our data, then you can say if you have this model structure, that the modeling of the data actually results in very well capturing the actual data. So 95% of the interval is kind of an indication of how well you capture the data. So this model structure does the job. But of course, we are not interested in what happened, not in the end, in what happened in animal brain, but we would like to know what happens in human brain. So what do? We take the system properties of the rat out and we plug the human properties in. So there you see the switch of the system. So then we have the same model structure, but we have different volumes, flows, and all these physiological things, but the structure remains the same. And with this, we can start to predict, okay, so we did. So here, if we have a plasma pharmacokinetic profile like in the black line, I hope you can see it, we would predict that we have brain extracellular fluid concentrations like this. And as another example, the subarachnoidal space, so the lumbar concentrations like this. And you see the distinct profile from this and this. Okay, predict, okay. And it is important just to see if this makes sense. So what we did is actually to have published data on what was measured in patients. This is actually healthy subjects. It's from 1996 from Banworth. I should have added the reference. Here you have the plasma pharmacokinetic data that kind of is indicated by the black dotted line. So that is an okay. And that is of course what we used as input because that is what the model uses. And then we had as a result indeed an overlap of what we predicted and the actually observed data. So we thought, okay, we are in business. So this is indeed a very important approach. But this is only one drug, one system combination, or 2 systems actually, the animal and the brain. So a lot of other data, multiple drugs that we used with distinct physicochemical properties to really start to understand if we do all animal work, what is the influence of the physicochemical properties of a drug. And that is what you can tease out if you keep the system the same and only vary the drug properties. So that is what we did. And a lot of people worked with that. So as a huge team, we worked on all kinds of combinations of drug properties and measured those. And then Yumi Yamamoto was the PhD student that actually came from the individual drug models that Joost Westerhout made for acetaminophen, but also for quinidin and for methotrexate. And she made a generic model out of it. So where we could kind of take all together, and if you use this model, it should be nicely able to model our data. And to hopefully convince you, you can see here plasma, brain extracellular fluid, CSF in the lateral ventricle, CSF in the cisterna magna, and here the same for different drugs. So acetaminophen, atenolol, methotrexate, low dose, high dose, with and without blocker, etc., etc. Morphine, low, high, phenytoin, paliperidone, risperidone, rimoxiclib, etc. And you can see our actually measured data as black dots, and the red line through is the model prediction there. And then you can see it nicely overlaps. It's so tiny, but this is, I think, a couple of years kind of condensed in one slide. So you can see it really captures all these data. So it means that the model structure is a good structure. Okay, and then as an example, what could we do? So if we would have, for instance, morphine data from actually human brain, and that's an exception. And in traumatic brain injury patients in Uppsala at that time, it was allowed to have microdialysis probes because lactate and pyruvate are measured, and that is actually of help for the patient. That should always be number one. But also they gave morphine, and they were allowed to measure morphine in these brain extracellular fluid samples. So you had traumatic brain injury patients, one contusion on one side, so you had a diseased part and a healthy part. Microdialysis probes were put in the diseased part and in the healthy part. And here you can see for these two patients the concentration time profiles in the dot, the black dots and the open dots. So the dotted line and the red thing is actually what our model would predict. And it's pretty close. It's pretty close. And then in the diseased part, we found drug concentration time profiles like this. And then our healthy human model, of course, does not capture the disease part because the disease makes changes in the processes. So the result will change in different pharmacokinetics. That was the first one. And then here, this is Naomi Ketaranathan, and she worked in Cape Town in children that had traumatic brain injury. And then there, the probe was only placed at the right side, the healthy side, that was allowed. And if we would have the plasma pharmacokinetic data here, we could predict actually in all cases very nicely what would happen in brain extracellular fluid of these patients. And there were two cases where it was not so good. Here we had actually too little pharmacokinetic data in plasma, and we could only take this part. And here, of course, we don't have information here, so that is a guess. So only 2 blood samples is too little, and in this case, this child actually had diffuse TBI, so the whole brain was traumatized. And then you get more disease impact. So with these data, we try to validate our model. Then the next step is, of course, so I'm kind of against working with animals unless you use them for a very good purpose and try to replace them. And that is what we did. So using this information, we kind of came up, and I won't go into details, we made a first version of a fully physiology-based pharmacokinetic model. And that indicates that we have only drug properties to be put in, to be combined with the physiological parameters that the model has, and then predicts what happens. And actually, yeah, again, predictions need for validation. We always should gain trust in a model like this. And here you see the results of just plugging in these physical chemical properties and actually the prediction of what has been observed in these animals. So you see in all cases, so the median and then the 90% interval outward. So you see in all these cases how that works. So we thought, okay, that is actually quite nice. And this is just a kind of abstract. So what we do, we have input in the model, and that is actually CNS system properties, and this is the human brain. We have drug properties, we have our model, and then we have our prediction here again, and we can overlay it with actually observed data. And here is the brain extracellular fluid, and here is the lumbar region for acetaminophen. This is for morphine, by the way, and this is for oxycodone. It was not part of the model development. This is out of this, so it's external data. So that was actually very nice. And then, just as a remark, so this is a generally applicable model, and we only used 230 rats to have this generic model and compare it in pharmaceutical industry, what they need in order to obtain information for individual drugs. So this is really smart, small data. Okay, and then Mohamed Saleh was the next PhD student, and he took it one level further, because now we used the pH of each individual compartment. And we could explicitly distinguish between the neutral and the charged drug molecule transport. And also we had explicit equations for tissue binding based on phospholipid-lipid composition of the brain. So that is included. And here you see the colors that Mohamed liked to use. He used his own color. So this is for plasma, brain extracellular fluid, and this is CSF in the lateral ventricle, and this is CSF in the cisterna magna. And then you can see again the data set that we have and overlaying that it looks very good. And this is the sparse human data that we have, and indeed is also a very good model. Then I think we can kind of trust the model. And the next step, what you can do is kind of see what if. You can explore what if scenarios. You don't need animals for that. You can first let your model do the job in order to understand what would be the impact of changing one parameter and how would that ultimately end up in how we interpret things. And one of the important things is actually, for instance, the impact of CSF flow and the impact of the volume of CSF. For instance, in Alzheimer's disease, we know the brain shrinks. We get relatively more CSF. Turnover is slower, etc., etc. So there are many things, and also you investigate that in a way. And what we found, keeping the plasma pharmacokinetics the same, we found that changes in CSF flow and also changes in volume do not influence the brain extracellular fluid, but they influence how the CSF profile looks like, indicating that CSF profiles are not good indicators of brain extracellular fluid per se. So that is important. And we did this for many compounds and we found the same pattern. So actually the relationship between brain extracellular fluid concentrations and lumbar CSF is CSF volume and flow dependent. Okay, then we scraped off literature in search of all kinds of changes that can occur in physiology due to important diseases like brain cancer and Alzheimer's, Parkinson's disease, etc., etc. And we looked, for instance, the tight junction space that can be of influence. We looked into the pH of the extracellular fluid. Most diseases change the pH, and also the pH of the intracellular fluid. And just looking at these things that could influence passive transport only, because many people look in active transport, we did the following. We changed the paracellular space, the pH in brain extracellular fluid and in intracellular fluid. And here you can see a panel for one drug, and this is brain extracellular fluid, and this is brain intracellular fluid, and this is changing the paracellular width, and this is pH of the extracellular fluid and the intracellular fluid. So you can see for these compounds, so this genestin, morphine and codeine, you can see that these compounds are quite sensitive to these changes. There are also drugs that do not change. For instance, phenytoin. Phenytoin doesn't care whatever happens. It is very predictable just from plasma onwards. But other drugs do. We should know when things can change. And this is just a heat map showing all kinds of physiological parameters in the model and different drugs and different things like the Cmax, the Tmax and the AUC. So that is typically pharmacokinetic parameters, and we see where things changed mostly. So these type of things, you can kind of explore the landscape and see, okay, this is what we should be concerned about, or maybe to have an animal experiment dedicated to investigate whether such change indeed leads to these changes. Okay, to combine for this CNS-PBPK model, so this actually will help in systematically unraveling and predict the CNS pharmacokinetics in multiple CNS compartments, because we also include, of course, the spinal cord CSF, and therewith also the relationship between plasma pharmacokinetics, brain extracellular fluid, brain intracellular fluid, and CSF in these multiple places, I should say. And we can understand how these relationships are actually affected by conditions such as CNS diseases, and how that then influences the target site concentrations and ultimately also link to the receptor occupancy and the effect. So we're trying to really go through this line that I always will present again and again. Good. Then we come to, for those who are not part of the QSPainRelief Consortium, I can shortly explain. We have many people here that are part of the QSPainRelief Consortium, and that is actually focused on the effective combination treatment of chronic pain in individual patients by an innovative quantitative system pharmacology approach on pain relief. And we try to integrate different models and information at different levels, also in vivo, in vitro, in vivo animal, in healthy subjects and in real-world patients in order to understand how this relationship works, ultimately to improve the profile for individual drugs. And we have Berfin, and she is here, and she works on the pharmacokinetic part mostly of this model at different locations, because chronic pain is actually different sites where many things happen. So that should be unraveled, and Koen van Hasselt is a co-supervisor in this. And then we have Divakar, and he's also here, and he actually works on the binding kinetics of multiple drugs at multiple locations in the brain. And also Koen van Hasselt works with this. So we will have a general assembly meeting from tonight onwards to further see where we are in this project. So, in conclusion, I think complex questions cannot be answered by simple tests. And then I would like to kind of emphasize the importance of smart data, connected data, because these are key to connecting pharmacokinetic and pharmacodynamic relationships and its translation to the human. And if we would have predictive models, that is the focus, we can also explore what-if scenarios, and that will help in better testing of molecules in the preclinical as well as in the clinical setting, to be more targeted to what you really would like to make sure. And ultimately also, of course, predictive models will help in reduction and ultimately replacement of experimental animals. So then I have the 1, 2, 3, 4, 5, 6, 7 R's now all together: right place, right time, right concentration, right person, and then the reduction, refinement and replacement of experimental animals. And of course this is a team approach. So here I depicted quite a number of photos of the people that have contributed to all the work. And thank you all, and if there are questions, I'm of course happy to answer them, because we will have a discussion setting. Thank you very much.